next topic, that is, I just told you that one more signaling pathway is important in cancer, that is NF kappa B. NF kappa B is deregulated in many cancers. We will look at it carefully. We have already discussed this, that is, the cancer cells have several hallmarks. All of them could be affected by a deregulation in NF kappa B. That is why this slide was brought in. Self-sufficiency in growth signals, insensitivity to anti-growth signals, evasion of apoptosis, that is what we are mainly focusing on, limitless replicative potential, sustained angiogenesis, tissue invasion. These are important acquired properties of cancer cells we have already looked at. And the tumor cells interacting with the microenvironment is also an important factor for them to become metastatic. This apoptosis, I have already told you how apoptosis is important in forming digits, forming lumen, losing the tail, adjusting cell number, different roles. So, this eliminating infected cells, there are many roles for apoptosis. If you remember that I just told you in the last class that NF kappa B plays an important role, anti-apoptotic role, when apoptosis is induced by different agents like TNF, radiation and chemotherapeutic agents. And I also told you that several papers have come in very good journals. These are the actual research publications. An essential role for NF kappa B in preventing TNF alpha induced cell death. As I told you, it came in Science in 1996 from David Baltimore, who is a Nobel laureate. Again, Baldwin, Albert S. Baldwin Jr. They have worked with the TNF and cancer therapy induced apoptosis. Again, potentiation by inhibition of NF kappa B, suppression of TNF induced apoptosis by NF kappa B, more or less same thing from Inder Verma's lab. So, all these labs are very famous labs, and all of them have shown this particular idea, which I already told you, that is TNF, radiation, chemotherapeutic agents, they induce apoptosis. At the same time, they activate NF kappa B, and that activated NF kappa B induces proteins which will block apoptosis. So, if you block NF kappa B, then you may be able to increase the efficiency of the anti-cancer agents to induce apoptosis. So, that is the idea. Now, we will look at what is NF kappa B. NF kappa B, as I told you, is a family of closely related dimeric transcription factors. The most important dimer is P50 and P65, that is often called classical NF kappa B. What they do as a transcription factor, they will bind a certain site in the DNA, right. So, they bind a common sequence of immunoglobulin gene called kappa B site. They were found in B cells, that is how, and in the kappa B region. So, that is how it was nuclear factor kappa B is the full form, held inactive in the cytoplasm by an inhibitory factor called I kappa B, and activated. This is one shortcut method to activate transcription factors. You remember I told you about this. P53 also can be activated like that, right. If you I mean, in that case it is inactivated by MDM2, but if you inactivate MDM2, you can activate P53. So, these are some simple methods by which transcription factors can be immediately activated without an elaborate procedure of synthesizing some proteins and things like that. So, here they are held inactive in the cytoplasm with the I kappa B's. When the I kappa B binds to NF kappa B, it becomes inactive. So, activation occurs when you degrade the inhibitory proteins. Then what happens? The NF kappa B translocates into the nucleus, binds to specific gene promoter, and then it promotes transcription like any other transcription factor. So, this is the nuclear kappa B signaling pathway, but there are many members. Two classes, you can classify them based on their synthesis and transactivation. Class 1 is usually synthesized in mature forms, that is rel A or P65. Rel is an oncogene because reticuloendotheliosis virus and so on. So, that is derived from that. Rel A is an oncogene actually. So, rel A P65, rel B and C rel. It is because rel homology domain with nuclear localization signal, this is important, and then transcription modulating domains. So, all these class 1 molecules are synthesized in mature forms, and they have rel homology domain with nuclear localization signal, and they have this transcription modulating domains. Class 2, the members are P50 and P52. P50 is originally synthesized as a large precursor. As you know, these numbers denote the molecular weight. P105, 105 kilodaltons. It is called NF kappa B1. It has to be cleaved to get P50. So, if you cleave P105, you will get P50. Similarly, P100 or NF kappa B2 has to be cleaved to get the mature form P52. The mature forms are P50, P52, P65, rel B and C rel. So, they are the mature, that is the large precursors from which they are formed. The interesting features are they have the rel homology domain and what are known as ankyrin repeats and ubiquitin dependent proteolytic cleavage required for processing. You remember I told you P105 has to be cleaved to get P50. So, that is done through ubiquitin dependent proteolytic cleavage into DNA binding forms P50 and P52. So, basically P50, P52, P65 all these things are DNA binding forms. Usually DNA binding forms are dimers. P50 and P65 will form a classical NF kappa B heterodimer. So, that is what we call as NF kappa B. There are other combinations as well. Now, there are these inhibitors of kappa B. As I again told you already, there are a family of proteins I kappa B alpha, I kappa B beta, I kappa B epsilon, I kappa B gamma and BCL3. They contain an amino terminal regulatory domain within which there are two conserved serines, because you need something to be phosphorylated. That if you phosphorylate these serines in the I kappa B, then that leads to ubiquitin mediated degradation. You need to degrade the I kappa B to activate NF kappa B, because they are bound as inactive complexes. You have to inactivate the inhibitor. If you inhibit the inhibitor, the NF kappa B gets activated. I often told you this is a mechanism. Now, you can look at it. This slide tells you all these things. That is how the rel P65, C rel, all of them have, see how many amino acids are there, all those details are given here. You can look at the ankyrin repeats, which are shown in a different color here, right. There are so many ankyrin repeats. Generally, the mature forms of NF kappa B, that is the P65 and all, they do not have this, whereas I kappa B's generally have this ankyrin repeats. These are some lower organisms also have certain homologs like Drosophila, dorsal, diff, relish, all these things. Then they have shown the rel homology domain and so on. So let us not worry too much about it. Now you may want to know what actually in the system activates NF kappa B. There are many conditions. Generally bacterial infections, Helicobacter pylori, Mycobacterium tuberculosis, lipopolysaccharide. The most important ones I have just because there are too many, I do not want you to memorize. So, I have shown the important ones in red color, lipopolysaccharides, bacterial lipopolysaccharides. Viruses or their products. There are a number of viruses, HIV, influenza virus, double stranded RNA viruses, HCV, hepatitis C virus, usually the core protein. Many cytokines can induce apoptosis, induce NF kappa B. Interleukins, many interleukins, 1, 12, 2, 15, 17, 18, interleukin 18, all these number of interleukins can activate NF kappa B, because basically NF kappa B is important in the immune system. During inflammation, NF kappa B gets activated. So, it is very important there also. Do not get confused when we are talking about cancer only. Here we are specifically focusing on the anti-apoptotic property of NF kappa B, but then it has got other functions as well. We will look at it briefly, but not elaborately, because we are looking at cancer. So, our aim is that. Many growth factors like EGF, epidermal growth factor, the simple serum also can. You can imagine when we are doing experiments in the lab, we are using the serum. So, that can also activate. This is a complication when you are assessing NF kappa B, because serum itself can induce NF kappa B. EGF, insulin, platelet derived growth factor, TGF beta, transforming growth factor beta, NGF, nerve growth factor. Radiation again is an important activator of NF kappa B, UV radiation, ionizing radiation. Stress, any stress can activate. Hydrogen peroxide, the chemicals like pervanadate. Anti-cancer agents, I already told you about this. Cisplatin, daunorubicin, etoposide, tamoxifen, Taxol. Many anti-cancer agents, they can induce NF kappa B. Here you realize the contradiction. The anti-cancer agents, they induce apoptosis, they also induce NF kappa B. That means the cell, this is a, you can understand it this way. The anti-cancer agents are trying to kill the cancer cells. The cancer cells, they do not want to die. So, it is a resistance mechanism. So, they produce more NF kappa B and thereby they can block that action. So, that is the idea. So, if you understand that, it is easier to. So, it is ultimately it all boils down to who wins the game, who is more powerful. If the anti-cancer agents are more powerful, they are going to induce apoptosis. Or if you want to help to kill the cancer cells, you add NF kappa B inhibitors along with the anti-cancer agents, so that this opposing force is mitigated. So, that is the thing, simple logics, these are used. That is how these 3 papers, Science and Nature, that they came. That was the logic behind those papers. So, chemical agents like N-methyl NMDA, N-methyl D-aspartate, calcium ionophores, Farbol esters, many things can activate NF kappa B. Now, you may wonder, as it is a transcription factor, what are its targets, right? There are more than, this is a complicated subject. NF kappa B may have more than 100 targets, 150 targets. There is another interesting thing I want to point out. The targets can be pro-apoptotic as well as anti-apoptotic. This will be slightly confusing because we have given a partial view that it is always anti-apoptotic. That is not the case. It is a transcription factor. It has 150 targets. Some of them can be pro-apoptotic, some of them can be anti-apoptotic. So, under some conditions it can be pro-apoptotic also. This is confusing, but in the cancer scenario we are talking about the anti-apoptotic thing. So, if it can have 150 or 200 targets, and if some of them are pro-apoptotic, if the condition is favorable, it can turn into apoptotic also, right. So, that you have to keep in mind. But generally, for a general understanding, the NF kappa B induced proteins are anti-apoptotic. That you remember that. Another interesting feature that you can note, TNF I told you is an activator of NF kappa B, and it can be a target of NF kappa B also. So, these are some certain things that you have to keep in mind. Something is inducing NF kappa B, and that NF kappa B again induces the production of something that induced it, here TNF. Similarly, some of the interleukins, they were activating NF kappa B, they are also targets. That means, this is one mechanism that can replenish the NF kappa B again and again, right. So, more and more NF kappa B could be formed. Of course, there are opposing mechanisms. Okay. Growth factors VEGF, cell cycle regulators, cyclin D1, P21, apoptosis regulatory proteins. This makes sense. BCL2 is an anti-apoptotic protein. So, that is one of the important targets of NF kappa B. Similarly, inhibitors of, this is very, very important, that is why I put in red, inhibitors of apoptosis proteins. So, now you can understand the mechanism. NF kappa B is responsible for the synthesis of inhibitors of apoptosis proteins, which block apoptosis. So, more and more NF kappa B, more and more inhibitors of apoptosis proteins. So, more and more blockage of apoptosis. That is the mechanism by which NF kappa B can block apoptosis. Because you remember, in inhibitors of apoptosis proteins, they can block caspases, right. I have told you, you must, you may remember the earlier slides I have just told you. Then there are COX2 proteins, INOS. Again, interestingly, the serial I kappa B alpha is also one of the targets. I kappa B is actually an inhibitor of NF kappa B, but it is also a target. This is a feedback regulation, it is a feedback inhibition. So, see, that is what in biology many things happen, many pro and anti apoptotic factors will be synthesized, but ultimately it is a war between these factors and something will win. So, you cannot be surprised with the end result, because it may not be as you expect. That is why this confusion comes in biology, because you expect something to happen, but it may not happen in the expected lines, because in the internal milieu, cellular milieu, something may happen. In the microenvironment, it might favor one condition or the opposing condition also. So, that has to be kept in mind. This is how we have to understand. Then we have to go and dig back what could have led to this condition, which we have not expected. The genes, a number of genes are regulated by NF kappa B. Of course, we are interested in proliferation, so we have concentrated on this. Anti-apoptotic factor we said, so that is done through upregulation of IAPs, inhibitors of apoptosis proteins, BCL2, BCL-XL, and this is so called A1 and C flip. These are all anti-apoptotic. NF-kappa B is also involved in proliferation. This is very interesting. It is not only that it can enhance survival by anti-apoptotic proteins, but it can also enhance the synthesis of oncogenes like c-MYC, cyclin D1. So, it is involved in proliferation. So, it is a two-pronged thing. NF-kappa B can promote proliferation and anti-apoptosis, and it is very important, although we are not discussing it in immunity. In immunology, it is very important, because it can be responsible for the upregulation of several chemokines, cytokines, antimicrobial peptides, adhesion molecules, INOS and COX-2. And last but not the least, it is also involved in the negative feedback of its own synthesis by promoting the synthesis of I-kappa B alpha, I-kappa B beta and A20. So, this is the interesting thing. That is why I told you several more than 100 genes are regulated by NF-kappa B. They have variety of functions like this. You can classify them under four categories: proliferation, anti-apoptosis, immunity and negative regulators of NF-kappa B. So, this has to be understood. Now, I have not told you the pathways, but these are the three pathways of NF-kappa B signaling. What are known as canonical pathways. This is a typical pathway. Canonical pathway I will describe by another figure. This is the simplest figure that we can look at, so that we will be, this is the actual NF-kappa B signaling pathway. How it happens? There are inducing signals. What are those inducing signals? I already told you. TNF, so many inducing signals, radiation, many inducing signals. What these inducing signals do? They impinge upon one complex of enzymes called I-kappa B kinase complex. There are three different I-kappa B kinases: I-kappa B kinase alpha, beta and gamma. Gamma is a regulatory subunit. So, basically they phosphorylate this I-kappa B kinase that gets activated, that now phosphorylates I-kappa B alpha. You remember I told you there are two serine residues at the N terminal of I-kappa B. If I remember, it is 32 and 36, you can verify that. I may not remember it exactly. What normally happens is, this is a very important one. Normally, if you remember, I told you NF-kappa B means P50 and P65. P65 is REL-A. It is a heterodimer. This is what you call as NF-kappa B. Now, this is kept inactive by binding with I-kappa B. Basically, what it does is, the nuclear localization signal is blocked by this binding. Now, these kinases, you remember, they get activated, they phosphorylate I-kappa B, and this phosphorylation is a signal for ubiquitination and consequent degradation in the 26S proteasomes. Now, I-kappa B gets degraded. Now, NF-kappa B is free, P50 and P65 are free. They move into the nucleus, they bind specific sequences and they transcribe those 150 genes that I told you. So, this is the canonical signaling pathway. This is what is shown here. For example, TNF receptors, then the I-kappa B1 and I-kappa B, the same as I-kappa B alpha and beta. There is one enzyme called NEMO, and they degrade I-kappa B, and that results in a number of target genes being activated. That is what is shown in canonical pathway. There are also what are known as non-canonical pathway. See here, if you look at the canonical pathway, the actual NF kappa B is P50 and P65. Whereas in non-canonical pathway, rel B and rel A, rel B and P52, sorry, rel B and P52 will form a dimer. So, there are different combinations. Instead of P50 and P65, that is a normal one. Here it will be rel B and so called P52. So, that is the dimer here. So, that is the difference. And also there are only these 2 kinases instead of those 3 kinases. Here there will be only 2 kinases, and the activating signals are different, LT, beta and BIFF. These factors can and it involves another kinase called NIC. So, this is called non-canonical pathway. There is yet another pathway which is called atypical, with some of the UV light and doxorubicin. We do not know what are the kinases that activate. P38 is said to be involved, and also casein kinase 2 is also said to be involved. We are not very clear about the atypical pathway, but then it also activates this pathway. P50 and P65 is the dimer, and then target genes are. So, there are basically 3 different pathways that tumor necrosis factor, for example, activates NF kappa B. At the same time, it also induces apoptosis, and activation of NF kappa B results in the induction of anti-apoptotic proteins which block the apoptosis. So, a better strategy will be to block NF kappa B together with the induction with tumor necrosis factor or radiation or chemotherapeutic compounds. So, NF kappa B I have just told you what are the various activators and targets. Now, let us look at, now you understand why we need inhibitors of NF kappa B, because they may be able to potentiate the activities of anti-cancer agents, TNF and radiation, right. So, that is why people started looking for NF kappa B. What is the biological inhibitor? We all know it is I kappa B. I kappa B is the biological inhibitor. Apart from that, several natural sources have also given rise to certain inhibitors, as you can see in this table. Biological inhibitors, pox virus decoy receptors, adenoviral E3 complex, Yersinia, adenoviral E1A and all that. Natural compound is curcumin also inhibits NF kappa B, and epicatechin from green tea also. A number of such naturally occurring compounds also can inhibit. Of course, when you say, generally when we say biological inhibitor, we mean the I kappa B, because it is present in the system. These inhibitors are listed under that because they are from natural sources. So, natural inhibitors you can say, or natural sources for the inhibition. There are also synthetic inhibitors like anti-TNF, what is commercially known as Remicade, soluble TNF receptor, Enbrel, IL-1 receptor alpha, some peptides, tirapeptide. So, these are also used as inhibitors of NF kappa B. There are also certain inhibitors which will actually inhibit IKK, which activates NF kappa B. So, in that sense, if you inhibit the kinase that phosphorylates I kappa B, that also is an inhibitor of NF kappa B. So, people have used that, thalidomide, aspirin and certain non-steroidal anti-inflammatory agents and so on. Proteasome inhibitors, because you know it is the proteasome inhibition that occurs when I kappa B is phosphorylated. So, that I kappa B is degraded and then NF kappa B gets activated because it is free to move to the nucleus where it can bind a specific sequence and activate transcription. So glucocorticoids are another example. So these are some of the examples. Generally, they work by either acting on I-kappa B phosphorylation and degradation, or inhibiting the IKK activation, or sometimes in the TLR receptor interaction, IL-1 receptor interaction, or TNF receptor interaction, and so on. These glucocorticoids and all, they work on the NF-kappa B translocation, the DNA binding. So these are all the targets where these inhibitors work. So you can basically design inhibitors which can interfere with different steps, either in the proteasome degradation, or with the IKK activation, or with the phosphorylation of I-kappa B, or with the translocation to the nucleus, and so on. So many people are interested in understanding or isolating, identifying natural and synthetic inhibitors of NF-kappa pathway. These three pathways I have already explained to you. Now, so what is the relevance to cancer? I already told you this kind of anti-apoptotic activity initiated by NF-kappa B would be advantageous to cancer cells to survive for a longer time. So accordingly, many cancers exhibit constitutive activation of NF-kappa B. Constitutive activation I already explained to you, spontaneous activation. We do not know what exactly activated, but you find excess activity in many cancer cells. So some examples are given here. Most often it is all because of defective I-kappa B alpha activity. Somehow I-kappa B is not able to inhibit NF-kappa B. What I mean by giving this example, like colon carcinoma, ovarian, pancreatic, thyroid, bladder, prostate, many cancers have shown increased NF-kappa B activity. In all these so-called solid tumors, there is no mutation. In fact, we have also in our laboratory tried that. I will be shortly explaining that. Whereas in hematopoietic tumors, there is I-kappa B alpha mutation. You do not see mutations in the solid cancers generally. That is one difference. Hodgkin's disease, acute ALL as we call, acute lymphoblastic leukemia, acute chronic myelogenous leukemia, CML we call, ALL and CML as we abbreviate them. Adult T cell leukemia, all these are hematopoietic tumors, blood cancers you can say in colloquial language. So in these things, sometimes they found some mutations. So this is what to be understood. Generally there is no mutation, but in certain hematopoietic cancers they found I-kappa B alpha